Assalamu alaikum everyone, and hello to all my dear amazing viewers. Yes, it's me after 3 long years. You all might have thought that this channel is dead. It was actually, but don't worry, I'm going to bring this channel back to life just like homeostasis brings our body back to life. So I'm back with this super interesting topic, that is homeostasis, the art of balancing the body, because our body just hates the drama. Now, me and you as humans, we might like the suspense, the thrillers, the heartbreaks, but our body, it doesn't. Our body has the internal organs like the brain, the liver, the kidneys, the lungs, the heart. They all just love the simple, constant, and the stable environment. So that is we are going to talk about today, that is homeostasis. And we will start this homeostasis. We are going to have the objectives in this session today. We will have the introduction to homeostasis, then we will talk about the components of homeostasis. We will talk about briefly about the importance of homeostasis. Then we will talk about the most importantly, that is what we are going to have the more focus on, that is the mechanisms of homeostasis. Then we will talk about the efficiency of homeostasis. We call that as homeostatic gain. And last but not the least, we will talk about the homeostatic imbalance and diseases. So let us start with the introduction of homeostasis. Now, homeostasis simply means maintaining the stable internal environment. Our body is made up of trillions of cells, and these cells have the environment inside our body. Now, that environment is called as the internal environment, and the constancy of that internal environment, maintaining the stable internal environment, is actually called as the homeostasis. Now, this term homeostasis was coined by Walter Cannon. And this Walter Cannon, he coined the term homeostasis in 1929, and he actually got this idea from the statement of another scientist, and that scientist was Claude Bernard. Now, Claude Bernard said, for a free and independent life, there is actually one condition for that, and that condition is the constant internal environment. So, the Claude Bernard stated that the constancy of the internal environment is the condition for a free and independent life. All right, now let us talk about the components of homeostasis. Now, components of homeostasis, there are three important components. The first is the receptor. Now, the receptor, whenever, for example, if we have to maintain a constant internal environment, for example, there is a change in the external environment, so our body will actually has to do something against that change. Now, that change will be detected by the receptors. That is the first component of the homeostasis. Just think about it, that your mother detects your dirty room. So your dirty room is the change, and that change is detected by your mother. So your mother becomes the receptor. Okay, we will look at the perfect examples in our body, but this is just for the understanding. So receptor is what detects the change. For example, your mother detects your dirty room. Now, this receptor will send this information. It will send this signal to the control center. Okay, now control center can be our central nervous system or the endocrine glands, because you all know that we have two major control systems in our body. One is the nervous system, second is the endocrine system. So the control system can be any endocrine gland, or it can be the brain or the central nervous system. Now, what control center does is, after receiving this information about the change from the receptor, your control center will make a decision. Your control center will decide what has to be done against this change. For example, in this example, what I have given you, your mother detects your dirty room, your mother will pass this information to your dad, your father, that father becomes the control center. Now, your father will decide what has to be done against the change, and that decision will be sent to the effector. And effector is going to be something who is going to execute the command that is coming from the control center. And in this example, you become the So in this example, you are going to be effector and you are going to execute the command that your father has given you. Okay. Now, effector in this case, when you receive that command from your father, you are going to clean the room. So the change was the dirty room, and the final result was you have cleaned the room, and it was completed by three components: the receptor, what detects the change, the control center makes the decision, and effector executes the command. And these signals, the receptor is sharing with the control center. These are called as the efferent signals, and the control center, from control center to effector, these are called as the efferent signals. Okay. Now, what is the importance of homeostasis? As I've already told you, that our body is made up of trillions of cells, and these cells are really very delicate. So they actually like that stable environment around them. They actually like that stable internal environment. So whenever there is no homeostasis, whenever this homeostasis is disturbed, that means your pH will not be maintained, your temperature will not be maintained, your blood pressure will not be maintained. What will happen? For example, your pH and temperature will not be maintained, the enzymes will fail to work. And if the enzymes will fail to work, that means our cells are not going to function properly. And if the blood pressure is not maintained, for example, there will be the fall in the blood pressure, and all these important functions are going to be disturbed if homeostasis is not there. So homeostasis jo hai, it is very, very important in our body. Now, what we are going to focus mainly on the mechanisms of homeostasis, how our body is actually regulating or maintaining this constant internal environment. Now, in the mechanisms, we have mainly two mechanisms what we are going to talk about. One is the feedback mechanism, and second is the feed forward mechanism. Now, when we talk about the feedback mechanism is further divided into two subtypes. One is the negative feedback, and second is the positive feedback. Now, let's understand the difference, and then we will talk about certain examples to understand it properly, this negative as well as the positive feedback. The negative feedback is whenever there is any change and our body is going to oppose that change to gain the normalcy. Okay, whatever the change is, our body is going to oppose the change. That is going to be our negative feedback. For example, our blood glucose drops, our body will try to make it high. Our blood glucose actually increases, our body will try to decrease it. Our body temperature increases, our body will try to decrease it. And our body temperature decreases, our body will try to increase it. So our body is going to oppose the change to maintain the normalcy. Then second is the positive feedback mechanism. इसमें अपोज नहीं होती चेंज, इसमें क्या होता है, द चेंज इज एम्प्लीफाइड, the change is exaggerated, and it is exaggerated until the final goal is achieved. We will understand this with the help of the examples. So positive feedback is where the change is amplified and exaggerated, but this exaggeration or this amplifying this change will be until the goal is achieved. The goal is achieved, it will stop. For example, during the childbirth, the oxytocin will be released, and there will be contraction in the uterus. We will see that in the examples. There will be contraction of the uterus. There will be release of oxytocin, and that oxytocin will induce more contractions in the uterus. Okay, and then during clotting, platelet will be activated. It will attract more and more platelets until that clot is formed. So these are the positive feedback and the negative feedback, the difference between the two. Now, let us understand this positive feedback and the negative feedback mechanisms with these examples. And the first, we will understand the negative feedback mechanism first. And the best example to understand is the glucose homeostasis. Okay, now for example, our blood glucose level goes up. Okay, our blood glucose level goes up because glucose has to be maintained in that normal range. If it increases or if it decreases, both are not good for our body actually. So if the blood glucose level goes up, what will happen? This increased blood glucose level will be detected by the receptors present in the pancreas. Okay, now this is called as hyperglycemia. Now if there is hyperglycemia, there are receptors, because we have to talk about the three components. So three components, the first is receptor, and in this case the receptor is present there in the pancreas. And I hope you all know that pancreas has got both the exocrine as well as the endocrine functions. Now, the pancreas detects the high blood glucose level or increased blood glucose level, and in response to this change, now the control center becomes the pancreas, the pancreatic islets, that is the endocrine part of the pancreas. Now the receptors are present in the pancreas, and the control center also is the pancreas. So pancreas will decide what has to be done against this increased blood glucose level. Now pancreas will secrete a hormone in response, and that hormone is going to be insulin. Now, why insulin is released? It is released in response to high blood glucose level. Now, what should be the job of this insulin? This becomes the effector. Now, what should be the job of this insulin? Its job should be to decrease the blood glucose level. Its job is, now insulin goes and tries to shift this blood glucose level to somewhere where it can be utilized. Now, this insulin goes to the body cells. It goes to the body cells and it acts on the body cells, and it stimulates these body cells to open these channels, and these channels are called as glucose transporters. And these glucose transporters, we call them as GLUT. Okay, GLUT is the glucose transporters. There are different types, but here you have to understand that this insulin will act on the body cells. It will stimulate the body cells to open these channels, and these channels are specially meant for the transport of glucose. And from blood, this glucose will be shifted into the cell where it can be utilized for the metabolic processes. So what we have achieved the goal. The change was increased blood glucose level, and the control center is the pancreas, who detected the change and then decided what has to be done against the change. It secreted insulin. Insulin went and acted on the body cells and made the body cells to open the channels, and those channels are glucose transporters. The glucose moves from blood into the cells and decreasing the blood glucose level. Now, opposite to this, if there is decreased blood glucose level, now what will happen? This decreased blood glucose level will also be sensed by the pancreas, the receptors in the pancreas. So this pancreas has the receptors, and this pancreas actually is going to detect the changes in the blood glucose level, whether it is high or it is low. Now, this low blood glucose level will be detected by pancreas, and it is going to It is going to stimulate the pancreatic islets, and in response to this low blood glucose level, our pancreas is going to secrete another hormone, and that is called as glucagon. Now, insulin was secreted in response to high blood glucose level, so it has achieved the goal. It has decreased the blood glucose level by shifting the blood glucose into the cells. Now what glucagon has to do is, this glucagon has to increase the blood glucose level. It has to find some source where it can get the glucose and take that glucose and put that into the blood to increase the blood glucose level. Now the best source is our liver. Now what this glucagon will do is, it will act on the liver. This glucagon will act on the liver, and we have glucose inside our liver in the storage form, and that storage form we call as glycogen. So glycogen is the storage form of glucose. So we have stored glucose inside the liver in the form of glycogen. Now what this glucagon will do is, it will act on the liver and it will stimulate the process of glycogenolysis. And what this glycogenolysis is, it is converting this glycogen into glucose. Now once this glycogen is converted into glucose, that glucose is released from the liver and that goes into the bloodstream, and we have increased the blood glucose level. So I hope you understood this, that this negative feedback mechanism is actually opposing the change. There was increased blood glucose level, and our body secreted insulin that decreased the blood glucose level. If there is decreased blood glucose level, the change was opposed by secreting the glucagon, and glucagon actually converts the glycogen into glucose and takes that into the blood and increasing the blood glucose level. Okay, I hope this example is clear. If it is not, let's understand the negative feedback mechanism with another example, but we will talk about it very briefly, because video should not be of the longer duration. So the another example is thermoregulation. Now, thermoregulation is temperature regulation. I've already told you that if there is a change, that change will be opposed in case of the negative feedback mechanism. Now, for example, there is increased temperature. There is increased temperature because sometimes the external temperature can sometimes go up to 45 to 50 degrees, and sometimes it can go to -10, but our body temperature has to be regulated somewhere around 36.2 to 37.2 degrees Celsius. So we have to maintain that body temperature inside. Now, for example, the temperature is increased. That increased temperature will be sensed by the thermoreceptors present in the skin. Thermoreceptors. Now these thermoreceptors takes this, these are the receptors. Now they take this information to the control center, and that control center is our brain, and the part in our brain that is the control center here is the hypothalamus. Because you all know that hypothalamus is very, very important for maintaining the temperature or thermoregulation. Now, what hypothalamus has to do, it has to, in response to that increased temperature, it has to cool us down. It has to cool the body down. Now, how it does this, how it is cooling our body down, it actually sends the decision. It actually sends the efferent signals to two different structures. The first is, we have the blood vessels going to our skin. We call them as cutaneous blood vessels, and that blood flow is the cutaneous blood flow. Now, you all might be knowing that our blood contains some amount of heat as well, and if this blood flow is going to the skin, we are radiating some of the heat out of the body through these cutaneous blood vessels. Now, what happens is, if there is increased temperature, if there is increased temperature, that means we want to radiate more and more heat from our body. Okay, we do not want to conserve the heat, we want to radiate the heat out of the body. So what will happen is, this hypothalamus, it will act on the cutaneous blood vessels and it will lead to vasodilation. And vasodilation means more and more blood flow will be going, and more blood flow will be going to the skin. That means more and more heat will be radiated out of the skin. The second thing is, it is going to stimulate these glands, and these glands are sweat glands. These glands, these are sweat glands, and these sweat glands will be stimulated and they will secrete sweat. And once the sweat glands secrete sweat on our skin, this sweat is definitely going to cool our skin down. So our hypothalamus acted on the cutaneous blood vessels, led to the vasodilation. Vasodilation, say cutaneous blood flow increased, we have radiated more heat out of the body, and then the sweat glands were also stimulated, and stimulation of the sweat glands lead to the secretion of sweat, and that actually helps us cooling down the skin. And then another thing is, we do not want to generate more heat in our body. So our hypothalamus, what it does is, it tries to decrease the muscle contraction, and muscle contraction will be decreased. That means less heat will be generated, so we are maintaining the body temperature inside our body. Now, in opposite to this, if there is decreased body temperature, the opposite is going to happen. This will be sensed by the thermoreceptors, it will be taken to the hypothalamus, and hypothalamus will do the opposite. It will, now in this case, it has to generate more heat, it has to conserve more heat. Okay, now what is going to happen is, in this case, hypothalamus is going to lead to vasoconstriction. Now, vasoconstriction, when vasoconstriction will be there, that means we are decreasing the blood flow towards the cutaneous blood vessels. We are decreasing the cutaneous blood flow. That means less amount of blood is going towards the skin. And if less blood is going towards the skin, we are radiating less heat from our body. So that is going to be the first thing. Then second thing is, it is going to inhibit the sweat glands. It is going to inhibit the sweat glands. That means there will be no secretion of sweat. And third thing, very important, it is going to stimulate the muscle contraction, and that is actually going to that is when there will be more muscle contraction, that is going to generate more heat. And that is why whenever it is very cold, we feel that shivering. That shivering is because our hypothalamus has triggered the muscle contraction there, and that muscle contraction is going to generate more heat inside our body and bringing back that temperature back to normal. Okay, so this is another example in the negative feedback mechanism. Now, when we talk about the positive feedback mechanism, the positive feedback mechanism is, the classical example is childbirth. Okay, now for example, during childbirth, what happens is that this fetus actually puts some stretch on the uterus or on the cervix. Okay, the stretch on the uterus and cervix actually stimulates these receptors, and these receptors are called as stretch receptors. Stretch receptors. And these stretch receptors will take this information again to hypothalamus. And what this hypothalamus will do is, it will send this information to posterior pituitary gland. And posterior pituitary gland will release oxytocin, and oxytocin will act on the uterus, and it will act on the uterus and it will lead to more contractions. Okay, now think about it. The change was contraction in the uterus, and because of that there was stretch on the uterus. And stretch on the uterus led to the activation of the stretch receptors. The information was taken to the hypothalamus, then to the posterior pituitary, more oxytocin, more contractions. Again, the same thing, more oxytocin, more contractions, and these contractions will go on until the child is born, the fetus is born, until this process of childbirth is achieved or completed. So this, in this case, it is not opposing the change, it is exaggerating the change until the goal is achieved. Let's understand this with the second example. Now, for example, let's understand here, for example, there is any injury. Okay, there is any injury, and that injury, actually there is damage to the endothelium. For example, here is the injury and here is the damage to the endothelium. And that damage to the endothelium will expose the collagen, will expose the extracellular matrix here, will expose the collagen, and what will happen is we have the platelets that has to stop bleeding there. For example, there is the injury to the endothelium, there will be definitely the blood loss. And what will happen is we have to stop the bleeding. We have to go for the process of hemostasis. Okay, not homeostasis, what we are talking about right now. We have to go for the process of hemostasis. That means we have to stop the bleeding. Now, what will happen is the platelets going on in the blood flow, the platelets will come and stick here. The platelets will come and stick here on the collagen or on the von Willebrand factor. That is the separate topic we will talk about in the hemostasis. Now here the platelets, once the platelet is coming here and it is sticking with the exposed collagen or the von Willebrand factor, this is called as platelet adhesion. Once the platelet gets adhered here on a non-platelet surface, the sticking of the platelet on a non-platelet surface is the platelet adhesion. So once the platelet adhesion is done, the platelet gets activated, and it releases certain chemicals, they will attract more platelets. They will attract more and more platelets, and those platelets will come and stick on the platelet surface now. That is called as platelet aggregation. And these platelets, once they get aggregated on the platelet surface, they will be activated, they will release the chemicals, they will attract more platelets. So this process goes on until the final goal is achieved. That means until the process of coagulation is completed, or the process of the formation of hemostatic plug is completed. One more time, we have the injury. There was one platelet, for example, coming in the area and sticking to the non-platelet surface here. Once that platelet gets activated, it exaggerates this thing, it activates and it releases the chemicals, and those attract more and more platelets. Once those platelets get activated, they attract more platelets, they attract more platelets. This process is exaggerated until the final goal, that is the formation of hemostatic plug, that is achieved. So this is another example of positive feedback mechanism. Now, there is something called as feed-forward mechanism. Now, feed-forward mechanism, our body is trying to compensate not after the change. Our body is predicting the change and doing something inside. Okay, for example, here is the example: the gastric juice secretion, even when the food is not in the stomach. Okay, the gastric juice that is secreted in the stomach, the gastric juice secretion, when we see the food, when we smell the food, when we think about the food, okay, the food is not even in the stomach, but our stomach is secreting the gastric juice. That is, it is preparing the stomach for that food that is going to come. So that is the feed-forward mechanism. Another example is, for example, you are going to do any physical activity. You're going to do any physical activity. Our body predicts that change. Our body predicts that during that physical activity, our body is going to demand more and more oxygen. So our body will, in advance, before that physical activity, what will happen? Our heart rate will increase, our respiratory rate will increase. That is the predictive control, and that is before the change has occurred. That is called as the feed-forward mechanism. Now, this feed-forward mechanism is kind of that intelligent version of the homeostasis, because it gets better over time. For example, the same example, the second example, we have to do any physical activity. For example, you have to climb the stairs. You have to climb the stairs. For the first time when you climb the stairs, you feel that breathlessness, because how much the body has predicted was not enough. But for the second time, third time, fourth time, you feel okay while climbing the same stairs. It is because our body is actually adapting to that change. Our body is adapting that. That is called as adaptive control. Our body is, this homeostatic mechanism is getting better over time. That is the beauty of this feed forward mechanism, not the feedback mechanism. Okay. Now, we will talk briefly about the homeostatic gain. Homeostatic gain is how efficiently the homeostasis is working. Okay? Now, let's understand this. This is the efficiency of the feedback system in correcting a disturbance, and the formula for this for detecting this efficiency of the feedback system is the correction divided by the error remaining. For example, there is any change, how much it is corrected divided by how much error is remaining there, that gives us the gain. That gives us the efficiency of feedback system. If there is high gain, that means there is tight control of the homeostasis. If there is low gain, that means there is sluggish correction. Now let's understand this with the examples. For example, we have to transfuse blood into a person. Okay, we have to transfuse blood in a person, and before transfusion, the normal blood pressure is 100. For example, mean arterial pressure is 100 mmHg. Okay, now if there are no baroreceptors, that means if this homeostatic mechanism is not there inside the body, without the baroreceptors, after the transfusion the blood pressure reaches 175. That means how much increase is there? There is 75 mmHg increase. Okay, please understand this. Before the transfusion, the normal blood pressure is 100 mmHg. If there are no baroreceptors, baroreceptors are the receptors that are detecting the pressure change. For example, if we are transfusing blood, that means we are increasing the volume of the blood. If we are increasing the volume of the blood, that is going to increase the blood pressure. That is straightforward. And that change in the blood pressure is detected by the receptors that are called as baroreceptors. Now, suppose baroreceptors are not there. Baroreceptors are not there. What will happen? Suppose this blood pressure goes from 100 to 175 after transfusion. And the change was plus 75 mmHg. Not gain, the change was plus 75 mmHg. Now, with baroreceptors, it is 125 mmHg. Now, what happens actually when we transfuse the blood? There is the increase in the blood pressure. It is detected by the baroreceptors. It sends the information to the central nervous system that tries to decrease the blood pressure during the transfusion or after the transfusion. Now, with baroreceptors is 125 mmHg. Now, how much is the change? That is plus 25 mmHg. Now, without the homeostatic mechanism, it is plus 75 mmHg, but with the homeostatic mechanism is plus 25 mmHg. Now, understanding the gain, that is how much is the correction? Correction is 50 mmHg. That means 75 minus 25, that means correction jo hui with the homeostatic mechanism, that is 50 mmHg, and we write it as minus 50 mmHg because it is the negative feedback mechanism. Okay, now error remaining is 25 mmHg, because without baroreceptors, this is a little bit tricky, but please try to understand that without baroreceptors, it was 175 mmHg, from 100 to 175. With baroreceptors, it is just 125 mmHg. So how much was the correction? It was minus 50 mmHg. 75 minus 25. Okay, and minus 50 mmHg, and error remaining is 25 mmHg, because the actual was 100 mmHg, but Even if the baroreceptors also acted, baroreceptors tried to bring the blood pressure back to normal, but it is still not 100, it is 125 mmHg. So 125 minus 100 is the error remaining, that is 25 mmHg. Now, applying that formula, what I told you is gain is equal to correction divided by error remaining, that is minus 2. Okay, so minus 2 is the gain. Now, when we see how much percent it is corrected, minus 2 is when we convert this into a percentage thing, it becomes 66%. Okay. Now, to understand this, for taking the percentage out of this, we have a formula: gain divided by gain plus 1 into 100. For example, in this case, what is the gain? Is 2. And gain plus 1 becomes 3 multiplied by 100. This actually becomes the 66%. So 66%, it is the moderate efficiency of homeostasis. Okay. Now, another example is, for example, the temperature regulation. Temperature regulation, what is the normal temperature? The average normal temperature is 37 degrees Celsius. Okay. Now, there is the disturbance of plus 2 degrees Celsius. Okay, there is the disturbance of plus 2 degrees Celsius, and because of that thermoregulation, it is corrected, and it is corrected to plus 0.06 degrees Celsius. That means plus 2 degrees Celsius was the change, and now the error remaining is plus 0.06 degrees Celsius. Now, how much is the correction? It is minus 1.94. It is 2 minus 0.06 is 1.94, and we write it as minus because I have already told you that it is the negative feedback mechanism. So how much is the gain? Minus 1.94 divided by plus 0.06, it becomes minus 33. And when we apply this in that percentage wala formula, it becomes 33 divided by 34 multiplied by 100, and it becomes somewhere around the 97%. So it is the high gain system, this temperature regulation. That means temperature regulation wala mechanism is more efficient than the blood pressure wala regulation. Okay, so because there the gain was 66% and here the gain is 97%. So this is the high gain system, this thermoregulation. Now, understanding the last example, for example, there is the sodium imbalance. The normal sodium is 140 milliequivalents per liter, for example, and it reaches from 140 to 150 milliequivalents per liter. And 150, that means change is 10 milliequivalents per liter. And after the kidneys detected, kidneys tried to maintain the sodium balance, sodium homeostasis, and they took it from 150 to 145 milliequivalents per liter. So how much is the gain? That is, the correction is minus 5 plus minus 5 and plus 5, that becomes minus 1. It's actually not plus, it is divided by 5. So this is the correction and this is the error remaining, because originally it was 140 milliequivalents per liter, and then after the homeostasis it was taken to 145 milliequivalents per liter. So that means it is the correction is minus 5 and the error remaining is also 5. So it becomes minus 1. Okay, and when we apply this in that formula of percentage, so it becomes the 50%. 50% जो है, it becomes 1 by 2 multiplied by 100. This 2 is 1 by 1 plus 1. Okay, so it becomes 1 by 2 into 100, it becomes 50%. So it is the low gain as compared to the other two. So we have talked about the three examples. The first was the pressure homeostasis, blood pressure, maintaining the blood pressure. That was उसमें जो gain percentage था, that was 66%. Then we talked about the thermoregulation. उसमें जो gain percentage था, that was 97%. अब we have talked about the sodium homeostasis by the kidneys, because kidneys are very slow to respond against this change. Okay, so kidneys are like, let me fix it tomorrow. Okay, so they are very slow. That means the gain system is very low. So gain, this is the low gain system, this sodium homeostasis. So these are the, for example, the homeostatic examples and the gain. And for example, very high gain is the body temperature. As we have already told, as I've already told you, that sodium balance is the low gain system. Okay, because there is slow renal response. Okay, then moderate में glucose regulation होता है या baroreceptors होता है. This is the moderate gain. Okay, now uh, we will talk briefly about the importance of homeostasis in health. Okay, or actually homeostatic imbalance we will talk about. If there is homeostatic imbalance, for example, there is the imbalance in the glucose homeostasis, that will lead to diabetes. If there is imbalance in the blood pressure reflex or disturbance in the blood pressure homeostasis, that will lead to heart failure. And if there is temperature regulation imbalance, that will lead to heat stroke. Okay, so these are some examples of the imbalance in the homeostatic system. So that is all for today. That is all for today in this lecture. I will see you in the next session. If you have any queries, please drop the question in the comment section. Thank you. See you.